Greetings, dear listeners. My name is Serikbayev Syrym, and today our broadcast is dedicated to genetic engineering. Genetic engineering, also called genetic modification or genetic manipulation, is the modification and manipulation of an organism's genes using technology. It is a set of technologies used to change the genetic makeup of cells, including the transfer of genes within and across species boundaries to produce improved or novel organisms. New DNA is obtained by either isolating and copying the genetic material of interest using recombinated DNA methods or artificially synthesizing the DNA. A construct is usually created and used to insert this DNA into the host organism. Genetic engineering has been applied in numerous fields, including research, medicine, industrial biotechnology, and agriculture. In research, GMOs are used to study gene function and expression through loss of function, gain of function, tracking and expression experiments. By knocking out genes responsible for certain conditions, it is possible to create animal model organisms of human diseases, as well as producing hormones, vaccines, and other drugs. Genetic engineering has the potential to cure genetic diseases through gene therapy. The same techniques that are used to produce drugs can also have industrial application, such as producing enzymes for laundry detergent, cheeses, and other products. Genetic engineering is a process that alters the genetic structure of an organism by either removing or introducing DNA or modifying existing genetic material in situ. Unlike traditional animal and plant breeding, which involves doing multiple crosses and then selecting for the organism with the desired phenotype, genetic engineering takes the gene directly from one organism and delivers it to the other. This is much faster, can be used to insert any genes from any organism, even ones from different domains, and prevents other undesirable genes from also being added. The DNA can be introduced directly into the host organism or into a cell that is then fused or hybridized with the host. This relies on recombinant nucleoside techniques to form new combinations of heritable genetic material, followed by the incorporation of that material either directly through a vector system or directly through microinjection, macroinjection of microencapsulation. Genetic engineering does not normally include traditional breeding. In vitro fertilization, induction of polyploidy, mutagenesis, and cell fusion, techniques that do not use recombinant nucleic acids or genetically modified organisms in the process. However, some broad definitions of genetic engineering include selective breeding, cloning and stem cell research, also not considered genetic engineering, are closely related, and genetic engineering can be used within them. Synthetic biology is an emerging discipline that takes genetic engineering a step further by introducing artificially synthesized material into an organism. And now our broadcast is over. Please subscribe to this channel. That was the host, Serikbayev Syrym. Goodbye.